In this video, we are going to study about Mendelian dihybrid cross. So here we are taking pea plant for the experiment. These are the parental plants showing two pairs of contrasting characters. You can see here the first parent is homozygous for red flower color and yellow seed color. And the second parent is homozygous recessive for white flower color and green seed color. As you know already, the dominant characters are represented by capital letters and recessive characters are represented by small letters. Here the parental plants are showing two pairs of contrasting characters. So the cross between these two plants are known as dihybrid cross. To make clear the concept, I have taken red strips as gene for red flower color and yellow strips as gene for yellow seed color. Similarly, I am using white strips for white flower color and green strips for green seed color. To do the experiment, I have taken 16 red strips, 16 yellow strips, 16 white strips and 16 green strips. Here you can see the genotype of the parental plants, that is red, yellow and white, green. The first parent is dominant for both the genes, so its genotype is capital RR, capital YY. The second parent is homozygous recessive, so the genotype is small rr, small yy. Next step is the formation of gametes. As we know from Mendel's law of segregation, when a gamete is created, it randomly gets one copy of each of these genes. Each character is represented by two genes, but in the gametes, each character is represented by a single gene. You can see here, in each gamete, one gene is from flower color and another gene is from seed color. Hi students, how many gametes are formed from the first parental plant? You can count and see. There are 16. By using the same pattern for the second parental plant also, we will arrange the gametes. Each gamete contains a single gene for flower color and a single gene for seed color. Here both the parental plants are produced 16 gametes each. The gametes from both the parents fuse to form the F1 generation. Here you can see 16 progenies in F1 generation. The genotype of all the 16 progenies are alike. The F1 individual is different from its homozygous parental plant. It is heterozygous in nature. That means the genotype contains both the dominant allele and recessive alleles. You can see here the phenotype of F1 generation, that is red flower color with yellow seed color. Because the red color is dominant over the white flower color and the yellow color is dominant over the green seed color. Next step is to cross these F1 individuals to raise the F2 generation. Mendel crossed two plants from the F1 generation. To raise F2 generation, let us suppose 8 clusters as male parents and the remaining 8 clusters as female parents. The first 8 4-strip clusters are considered as male parent. Here the parental phenotype is red-yellow. The remaining 4-strip 8 clusters are considered as the female parent. I hope you have noticed the genotype of the male parent, which is heterozygous in the flower color gene and also heterozygous in the seed color gene. Similarly, the female parent is also heterozygous in flower color and heterozygous in seed color. We have already learned law of segregation. As per that, the pair of alleles separate from each other during gamete formation, so that only one allele will be present in each gamete. Mendel's law of independent assortment tells us the alleles of different genes segregate independently. Here, this male parent, whether it contributes a gene for red flower color or white flower color, is independent of whether it contributes gene for green color or yellow color. So you have all of the different combinations that each of them can contribute. At the end, 4 different types of gametes you will get from the male parent. Similarly, the female parent also follow the law of independent assortment and will get 4 different types of gametes. They are red yellow, red green, white yellow, white green. Gamete from the male parent and female parent fuse to form F2 generation. At the end of the process, you can see here 16 F2 individuals. Next, we will arrange it based on the phenotype. As we know, red and yellow characters are dominant. The clusters which is having red and yellow stripes will arrange first. Next, we will select the cluster which is having at least one dominant character. Here in these clusters, red character is dominant. All the rest of the characters are recessive. In these clusters, only yellow is dominant. The last cluster is having only recessive characters. Here you can see 4 different types of phenotypes. They are red yellow, red green, white yellow, white green. The first 9 individuals are having the phenotype of red flower color with yellow seed. Next 3 individuals are showing the phenotype of red flower with green seed. Next 3 individuals are showing the phenotype of white flower with yellow seed. The last one and only individual showing the phenotype of white flower with green seed. The phenotypic ratio of the dihybrid cross in the standard Mendel experiment is 9:3:3:1. We have proved the same here. A dihybrid cross has a genotypic ratio of 1:2:2:4, 1:2, 1:2:1. Here you can see the genotype. You find it out whether it is correct or not. Thank you.